Good afternoon. Welcome everybody. I would like to thank the moderator of this session, and I would like to thank the organizing committee for giving me the chance and inviting me to be with you today. My topic will be the role of nutrition in brain development, the golden opportunity for the first 1,000 days. As regarding the brain growth, the perinatal period is the time of the most rapid brain growth for humans. It will continue to grow and mature at a rapid rate during the first 12 months of life. At birth, the brain has all the neurons it will need for life. Between birth and age 2 years, children will go through several stages of cognitive development, including sensory and language development. At term, human brain weighs 350 grams, reaching a weight of 1,000 grams at the end of the first year. For full-term, infant brain is 25 of its adult weight at birth. Age of 2, the brain will be 75. Age of 6, the brain will reach 90% of its adult weight. There are 5 stages of brain development: formation and closure of neural tube, nerve cell multiplication and differentiation, nerve cell migration, nerve cell maturation, and development of myelin sheath. Early nutrition and brain development will have an idea about the nutrient and growth factors as they regulate the brain development during prenatal and postnatal life. Rapidly growing brain is more vulnerable to damage, more amenable to repair following nutritional deficiencies. Different brain regions have different developmental outcomes. The factors affecting brain development are 3 factors mainly: the genetic potentiality, the environmental stimulation, and nutrition. All play together in the process of brain development. Any positive or negative effect of a nutrient deficiency or excess can affect the brain development depending on the timing, the dose, duration of exposure, and the momentary need to that nutrient. And effect of nutrition on cognitive development, as nutrient providing the building blocks that play critical role in neurotransmitter, hormone metabolism, important constituents of enzyme systems in the brain, cell proliferations, and DNA synthesis. As regarding undernutrition and brain development, there are animal studies in rats proved that malnutrition in rats appear to affect the cell number, cell migration, myelination, synaptogenesis, hippocampal formation, and neurotransmission. While in human studies, in severely malnourished infants, researchers described fewer number of neurons, shorter dendrites, abnormal dendritic spines. It's very clear in the picture. Moreover, malnourished children have less energy and interest for learning that negatively affects the cognitive development. Not only undernutrition affects the brain function, but also the overnutrition could affect the cognitive brain functions. In animals, there is a link between obesity and impaired cognition. Rats fed a high-fat diet, leading to decreased neurogenesis in the hippocampus. In humans, there is association between increased body mass index and reduced visuospatial cognitive function in 8 to 16 years old children. Also, the intrauterine growth retardation could affect the brain development and the cognitive functions because intrauterine growth retardation may affect the verbal outcome, the attention, learning, and academic achievement. 6.8 point IQ deficit at 7 years, 15% with mild neurodevelopmental abnormalities. So, in undernutrition, energy deficiency will lead to smaller brain volume and lower mental and psychomotor developmental scores. Malnutrition could affect structural growth and function development. structural growth in the form of cell numbers, cell migration, myelination, synaptogenesis, hippocampal formation, neurotransmission, and function development as interactive behavior and cognition. This is very important. So malnutrition could affect the main cognitive functions of the brain. Now coming on the nutrients that largely affect early brain development, we could divide it into three groups: the macronutrient as protein and fat, especially L-CPUFA; the micronutrient as iron, zinc, copper, selenium, and iodine; vitamins and cofactors like vitamin B, mainly B6 and B12, and vitamins A, C, D, E, with folic acids and choline. Coming to proteins. First of all, why the brain needs protein? For mainly four functions: synthesis of DNA and RNA and maintenance, neurotransmitter production, growth factor synthesis, structure protein as neuronal extension or synapse formation. The importance of proteins: as the diet-derived amino acids are essential for synthesis of structural proteins, enzymes, neuropeptides, and neurotransmitters. The essential amino acids, as we all know, include threonine, valine, leucine, isoleucine, lysine, tryptophan, phenylalanine, methionine, and histidine, arginine, and cysteine, which are essential mainly for low birth weight infants. The protein restriction can reduce the availability of these essential substrate, thereby affecting the brain growth. For protein energy malnutrition, moderate to severe protein energy malnutrition before 2 years is known to be associated with delays in cognitive development and school performance and decreased ability to learning. More recently, behavioral and cognitive deficits have been reported in relation to mild undernutrition. Coming to the second macronutrient with the fat, why the brain needs fat? It is a major source of energy. It provides essential fatty acid as linoleic and linolenic acids. Phospholipids and cholesterol serve as structural component of the cell membranes, important for formation of myelinated nerves. The brain is a lipid-rich organ. We should know that 2/3 of the brain is formed of fat. Complex lipids such as phospholipids and gangliosides comprise up to 35% of the total lipids in the brain. These complex lipids are critically important for the optimal development of the brain and visual functions. Regarding the long chains polysaturated fatty acids, polyunsaturated fatty acids, L-CPUFAs are the fatty acids with chain length more than 20 carbon atoms. 4 are particularly important for brain growth and brain function, which are the omega-6 fatty acids, arachidonic acids, and the omega-3 fatty acids, DHA, APA, and DGLA. Coming to the role of DHA as a structural component in the membrane for both the brain and the eye. It represents 97 of the omega-3 fatty acids in the brain and 93 of the omega-3 fatty acids in the eye. DHA plays a key role in photoreceptor growth and visual development. The importance of L-CPUFAs and supplementation in the prenatal stage has positive impact on the cognitive and visual function of the baby. L-CPUFA improves the scores on learning and processing of information tests. It also makes a high contribution to development visual functions as measured by the evoked potentials. So, L-CPUFA, especially DHA, may affect the development of the frontal cortex, responsible for visual acuity, language and cognitive development, learning and planning abilities, control of attention, and improving of memory. Coming to the second part is the micronutrient with the brain development. First of all, the iron. Why does the brain needs iron? For energy as iron found in cytochromes that make ATP, and the ATP utilization high in fetus and neonate. Neurotransmitters, as iron needed to make dopamine, serotonin, and norepinephrine. For myelin formation, iron-containing enzymes to make the fatty acids in the myelin. Iron is accelerated rapidly by the fetus during the last trimester and is necessary for basic neuronal processes such as myelination, neurotransmitter production, and energy metabolism. Iron deficiency anemia during the first 2 years of life is associated with poor performance on tests of intelligence or specific cognitive process at or near school age. Also, iron deficient infants have impaired auditory cognition and memory processing, whereas iron deficient premature infants have a higher rate of abnormal neurological reflex at 36 weeks post-conception. Very important that iron deficiency anemia could produce long-lasting neuropsychological effects and poor performance later in life. And there are many studies in human infants suggest that this is an irreversible effect that may be related to changes in the chemistry of neurotransmitters, organization, and the morphology of neuronal network and neurobiology of myelinations. So, protein energy malnutrition and iron deficiency anemia early in life lead to compromised growth and cognitive functions in young children. Furthermore, several studies have shown that iron deficient anemic children demonstrate delayed motor skills. And actually, this is very important, especially in the developing countries, as iron deficiency anemia and malnutrition is very prevalent. So iron deficiency anemia is one of the most prevalent micronutrient deficiency in young children in the world and strongly associated with persistent cognitive and motor delay even after the anemia and iron deficit have been repaired. It is reported recently that children aged 6 to 23 months with moderate to severe iron deficiency anemia had lower mental and psychomotor scores. These children also had lower scholastic achievement and needed more special education assistance than iron sufficient children. Coming to the second micronutrient, which is zinc. Zinc is essential for neuronal maturation and proliferation in brain functions, as it appears to induce the release of neurotransmitter GABA, gamma-aminobutyric acid, and serotonin, and thus affect the neuronal excitability and modulate the synaptic transmission. Severe zinc deficiency can cause retarded brain growth and maturation, abnormal cerebral function, impaired behavioral and emotional responses, and also may be related to anxiety and depression. In children, zinc deficiency has been associated with hyperactivity and sleeping disorders as well as the mental retardation. Zinc supplementation resulted in improvement of the quality of motor development and beneficial effect and impact on neuropsychological processes. Zinc supplementation has also found to be helpful in treating and curing postnatal depression for the mothers. Coming to the iodine, consequence of iodine deficiency is cognitive impairment, learning speech deficit with psychomotor problems. Iodine is needed for synthesis of thyroid hormones, which is critically important for normal brain development. And studies have demonstrated that it increases the proliferation of brain cells and regulates other processes involved in the brain functions. And iodine deficiency is the most common cause of preventable mental retardation and learning disability. Iodine deficiency in early life results in cretinism, which is characterized by mental retardation. And this is very important because actually this iodine deficiency and the sequence of mental retardation is preventable. So we could add iodine efficiently and do thyroid screening at birth to prevent mental retardation, especially the treatment is easy and not costly with thyroxine. Coming to the choline, the role of choline in the brain is very important as the essential nutrient for human substrate for the neurotransmitter acetylcholine, likely has epigenetic effect, promote larger neuronal size, more dendritic arborization, and greater neuronal signaling, especially in hippocampus, which is responsible for learning and memory. Folic acid is very important. The incidence of neuronal tube defects is definitely higher in children of women who are folate deficient during pregnancy. And low CSF folate levels among infants who started to have irritability, decelerating head growth, psychomotor retardation, cerebral ataxia, dyskinesia, and convulsion at age of 6 months, in spite of normal development before, patients were brought back to their normal state just with folate therapy. Copper. Copper is an essential role, has an essential role in brain function, as it acts as brain stimulant, improve epinephrine, norepinephrine, and dopamine production. Copper also increases the iron assimilation, so copper and iron work together in the formation of hemoglobin and the red blood cells, and thus help oxygen supply to the brain. Copper deficiency was associated with development of depression and auditory hallucination that improved with copper supplementation and recurred after stopping treatment. Copper deficiency leads to impaired motor function, impaired balance, impaired coordination. Coming to the vitamins, and the most important is vitamin B12, and infant born with vitamin B12 deficiency have hypotonia, involuntary muscle movement, apathetic look, cerebral atrophy, demyelination of nerves. While children with vitamin B12 deficiency will have lower intelligence, spatial ability, short-term memory. Coming to the magic of vitamin D, as it enters in every function in the body, and the consequence of vitamin D deficiency as regarding the brain, including cognitive dysfunction and autism, stroke, epilepsy, and in older age, Alzheimer, schizophrenia, depression, multiple sclerosis. For vitamin E is the parent compound of retinoids, signaling mechanism involved in homeostatic component of sleep electroencephalogram. Vitamin E is critical to hippocampus and hypothalamus, which control memory and wakefulness, plays central role in increasing neuroplasticity and neurogenesis, also playing an essential role in helping us in good quality sleep and long sleep long enough. For vitamin C, it is proved to be antioxidant and protective antioxidant effect for the brain cells, and important for collagen synthesis, involved in neurotransmitters production, mainly serotonin, and increase iron absorption. For vitamin E, which is powerful antioxidant, immune booster, cholesterol reducers, and skin protection. In conclusion, the seriousness of malnutrition is not only due to its effects on child morbidity and mortality, but also due to the possible effects on brain growth and development, which may not be totally reversible after nutritional rehabilitation. Examples, the iron. Great effort should be given for early detection and prevention of malnutrition through nutritional supplementation and the monitoring of growth and development of infants and children. And thanks for your attention.